Welcome to week 7 in the IES seminar series entitled Environmental Problems and Solutions in Small Island Developing States. Like I said, this is our 7th week. This week we have Dr. Sheena Francis from the University of West Indies, Mona, in Jamaica. Her title of her talk today is Trends in Vector-Borne Diseases in the Caribbean. Before she gets started, just a little bit about Sheena. Sheena is a research fellow at the Natural Products Institute at the University of West Indies, Jamaica. She mainly works on insect toxicology. Her work strives to understand mechanisms of insecticide resistance in Aedes aegypti, as well as to understand the behavior of the vectors. The goal of her research is to identify novel methods of vector control that can be used to reduce the transmission of mosquito-borne diseases by decreasing human mosquito contact. So there's a little bit of background about yourself, Sheena. So thank you for joining us today, and we look forward to your talk.
Okay, thank you everyone for having me. I do appreciate it. So the internet has been a little bit on the choppy side, so I'm going to turn off my my camera. So hopefully the presentation will go smoothly. All right, so I am aware that this method of giving a talk is not interactive. I can't really hear you, but I'm going to ask you some questions, and I would like if you could answer it in your own space. So question 1, have you ever traveled to a country and on returning home, and a health official or the customs officer asks you, have you ever been bitten by an insect on your travels? Have you, or question 2, have you or any family member have been infected by a disease transmitted by mosquitoes or ticks? So if the answer to those questions were yes, then you already know about vector-borne diseases, and you already know that all it takes is for one infected or viremic individual to travel to another country to start the cycle of transmission. So I see I have a question as a, oh, you weren't not asked those questions. Okay, then. All right, so my talk today will be looking at vector-borne diseases, particularly insect vector-borne diseases. And I'll be looking at trends in the mosquitoes and also including other organisms as I go along. And I'm looking at mosquitoes because, as said in the introduction, that's the insect that I work with. So majority of the information that I'll be presenting today is from a recent publication that I wrote, Challenges, along with other colleagues, Challenges in the Control of Neglected Insect Vector Diseases of Human Importance in the Anglo-Caribbean. God. Okay. So for the Caribbean, the English Caribbean to be specific, so I'm talking about countries that were under English rule at one particular time. Those are the countries that I'll be zoning in on. And then I'll be making references to other countries. So for the English Caribbean, the major insect vector that is important to us are the mosquitoes. And these mosquitoes specifically are the Anopheline mosquitoes that are known to transmit malaria, as well as the Aedes mosquito. And we have several types of Aedes different Aedes species circulating in the Caribbean. However, the Aedes aegypti tends to be the most competent vector that is important to us here. So the graph here displays mosquito-borne illnesses from 2000 to 2020 that's been reported in the English Caribbean. And this information came from PAHO on their PLISA website. So the orange bar shows the profile of malaria throughout the Caribbean. And malaria was introduced in the Caribbean in around the 17th century. And since then, many Caribbean islands, many English-speaking Caribbean islands have been able to eradicate malaria and are no longer required to report it. It's to WHO or PAHO. However, other countries such as Guyana, which is closely associated with the South American border, were unsuccessful in their eradication program. And of course, they are still troubled by malaria. So majority of the cases, the orange bars, are reports of malaria coming from Guyana. Now, other countries from time to time, through the importation of a person coming into the country with already infected with malaria, may also add to the contribution of malarial cases, but for the most part, those incidences are infrequent. Majority of malaria cases reported in the Caribbean is specifically owed to Guyana. The other insect vector, as I mentioned to you before, that's important to us are the Aedes species, particularly the Aedes aegypti. Now, the Aedes species prior to 2014 were only responsible for transmitting dengue in the Caribbean. That's the only disease that we had to worry about. And though there have been several eradication projects, we have been unsuccessful in eradicating dengue. And what we noticed throughout the years is that between the period 2000 to 2010, the dengue epidemics that the region observed was roughly every 5 to 10 years we'd observe an epidemic. Since 2010, the years between epidemics have decreased. We're seeing an epidemic now every 2 to 3 years, which is worrying. The other thing that we noticed in the Caribbean is that in 2014, one viremic individual prior to 2013 came to the French Caribbean island of Martinique. And because of the abundance of Aedes, and you can look at my map on the right-hand side that shows the abundance of Aedes aegypti throughout the Americas in general, which includes all of Latin America and the Caribbean. Red being the highest number, we have an abundance of Aedes aegypti here. And Aedes aegypti is a competent vector at transmitting the virus chikungunya. So all it took was for one viremic person to come to travel to Saint Martinique, a French Caribbean country, and be bitten by a mosquito there. And the local transmission of chikungunya started in Martinique. And because of travel and trade throughout the entire region, and the region depends on travel and trade, we are very dependent on tourism, the virus was traveled through the entire Caribbean within the space of a year. Similarly, in 2016, we saw the introduction of Zika virus throughout the region. Zika was introduced by a viremic person traveling to South America. Local transmission started there. And then, of course, Zika was observed throughout the entire region. Now, note on the graph that the incidences of chikungunya in its introductory year compared to Zika in its introductory year, 2014 for Zika, for CHIKV in its introductory year, 2016 for Zika in its introductory year, there's the incidences of Zika in comparison to that observed for CHIKV was very low. And that's primarily because at the time, in all honesty, Zika came in on the heels of the chikungunya virus. And many countries were well into their vector control program to try and decrease the population of the vector. We hardly have any medications out there to treat mosquito-borne diseases. They're still in their developmental stage. So our first line of defense for treating or dealing with mosquito-borne illnesses is population reduction of the mosquito. So many countries were well into their population reduction stage at the time Zika came in. Also, they were aware of the symptoms that were being observed with Zika. In that, in Brazil, there were reports of pregnant women being passing on the virus to their unborn child. So other government officials got involved. So we had aid from USAID in their Zika AIDS program that came to the region. We had PAHO, CARFA, and other activities going on to assist vector control programs locally in the Caribbean to reduce their vector problems. So with that, intensive vector control programs were able to decrease or suppress mosquito population to the point where we reduce the potential possibility of a human being infected by a mosquito. Now, with all of that said, mosquitoes are indeed difficult to control. Vector control programs have to be very intensive in order to control mosquito population, because the mosquitoes have become efficient anthropophilic vectors. Now, what do I mean by that? The mosquitoes are readily breeding in containers. So plastic containers, we normally have them for our convenience. They use them to package food, to store water in, and to transport goods. But sometimes we discard them in and around our homes. So the mosquitoes have utilized those refuge and made them their home. So they're breeding in close proximity to us. Because of the containers, the mosquitoes can breed regardless of wet or dry season. They only need 1 teaspoon of water to surviving. The mosquitoes also go through a period of diapause in hard conditions, meaning that, so the mosquito has an aquatic lifestyle that's egg to the pupation stage. So they go egg, larvae, pupa, and then adult stage is terrestrial. So they depend on water for their juvenile stage. Now, if conditions are unfavorable, if the mosquito is not getting enough water for its life cycle, it will go through a stage of inanimation. For example, the egg of a mosquito can survive 3 months without water. The very first stage of it, larval stage, can survive weeks without water. And as soon as water returns, it can resuspend itself. With this, mosquitoes can be transported easily by an unsuspecting traveler because they don't need water to travel. So they can move from one area to the next without being suspected. And with this, you have introduction of species. Now the Caribbean is seeing the introduction of the Aedes albopictus, which is a vector that was observed primarily in Asia. So some Caribbean countries are now reporting that they do have Aedes albopictus, and it's a more aggressive Aedes species than the Aedes aegypti. So in addition to diapause and ease of travel, container breeding, and their ability to survive in only a teaspoon of water, so they don't need much, they also have increased their metabolic rate so that during high ambient temperature, their rates of development from egg to adult has decreased. For me in the lab, I'll give you an example. I've noticed wherein from egg to adult stage in the summer times takes roughly 4 to 5 days, while in the rainy, cooler time, that takes roughly 9 to 10 days. So we have in summer months, we have a shorter time to development and thus more mosquitoes in the population. The mosquitoes are also responding to our habits. So once upon a time, they used to bite us in our home. So they used to be endophilic, loving the indoors. But now we're spraying inside the homes and we're trying to get rid of them. So they're like, okay, fine, I will move outside and I'll bite you outside. So they've now become exophilic. Their biting times have even varied. They know that we're most active in the day, so they're starting to bite us more in the day and less at night because they can get to us. And on top of all of that, they've become resistant to insecticides. As I've stated before, the mosquitoes have become resistant to insecticides. And this is really important because the very, very first line of defense to prevent mosquito-borne infection is in the use of insecticides. That's what our vector control and health officials use to decrease mosquito population. And they have, through the thickening of their cuticle over time, the thickening of cuticle prevents the insecticide from penetrating the skin, penetrating through to the skin and into the mosquito, preventing the mosquitoes from dying. The other thing is that they have increased the amount of metabolic enzyme activities in their body. Metabolic enzymes are found in all organisms, and the main purpose of a metabolic enzyme is to break down toxins or break down substances that come into the body. So the mosquitoes have increased their repertoire of metabolic enzyme to prevent the insecticides from getting to its target site to kill the mosquitoes. And if that is not enough, the mosquitoes have mutated their target site so that the insecticides that were designed specifically to bind to a specific target site is no longer active. They have altered or mutated their target site to prevent the exact binding of insecticide with target sites. So the insecticides have little to no use on these mosquitoes. They are ineffective mainly. So all of this makes population control in mosquitoes extremely difficult to vector workers. Now, we've started using models to assist with mosquito control. And I think Roxanne will talk about this more later down in the series. But just to introduce it, so there are trends. There are trends in the rainfall. Of course, the mosquitoes, as I said before, they have a juvenile stage that needs water. So the juvenile stage, you would expect that in rainy season, mosquito proliferation will be high, and in drier season, mosquito proliferation will be low. Also, you would expect that with an increase in mosquito population, you'll have an increased incidences of infected mosquitoes biting individuals, and of course, the transmission of vector-borne diseases to increase. So, so there's a trend, and countries are looking at these trends to assist them in their vector control activities. However, as I said, mosquitoes are highly efficient. They've become drought resistant. Now I want you to look at these two graphs. This is a paper that was published by Barrera and studies taken from Puerto Rico in 2011. Now the left graph shows abundance of pupae in containers in dry season, and the right graph shows abundance in pupae in the same containers in the wet season. And just at a quick glance, just look at it. Do you see any significant difference in the abundance of pupae in these containers? For me, the answer is no. So because of our habits of discarding containers, mosquitoes are very efficient at populating these containers. These containers, they get water. All they need is one teaspoon to survive. And with that, they are surviving irregardless of wet or dry season. So they've become drought resistant. Another thing to note, as I said before, is that the mosquitoes have become insecticide resistant, and insecticides are our first line of defense in controlling these populations. So globally, a study carried out by Moyes et al. In 2017 reported the countries that have reported insecticide resistance in Aedes aegypti between the period 2006 to 2015. So many countries in South America have reported insecticide resistance. But based on the graph, we're here, and you hardly can see us, but we're here. Not many Caribbean countries have reported insecticide resistance between the period 2006-2015. And this is concerning, considering that insecticides are our main line of defense. And we need to find out whether these insecticides are working and whether the mosquitoes are responding. So there's inconsistency in our vector control program at looking at what's out there and what we're doing. And I'll use Jamaica as an example to describe this. So in 1964, it was recorded that there was resistance to DDT in mosquitoes. And then 30 years after that, a study carried out by CAFA looking at mosquitoes sampled from all throughout the Caribbean, including Jamaica, found that there was relatively low malathion and temiphos resistance. And then in 1999, Castle et al. Looked at the application of the insecticides, fogging, and found that application to be ineffective, but we're still using it because it's relatively cheap. And then, of course, later on, my studies came along to look at insecticide resistance to commercially available insecticides in 2017 to 2021. So there's inconsistency in us looking at our tool, our main vector tool, and say working yes or no. But this could be in terms of publication, and maybe they're doing it at the local level and just probably not publishing the information, not sure. But for my initial study, I looked at mosquitoes that were collected from St. Andrew, Jamaica. And I'll tell you a little bit about my observation. So what I found was that less than 20% of the population sampled from St. Andrew, Jamaica were responding to the specific concentration of permethrin used, which is the diagnostic dose to test resistance. At the time, in 30 minutes, the diagnostic time to test resistance. A little than 20% of them were being killed by permethrin at the specific concentration in comparison to a known susceptible species of mosquitoes. The other thing is that the mechanism of resistance being observed in the population varied. So I sampled 5 areas throughout St. Andrew, Jamaica. And the bars, they represent target site mutations, the percentage of frequency of the gene in target site mutation. And the line, the black line, represents the metabolic enzyme activity. So note that for populations that had a high frequency of the target site mutation genes had a low profile of metabolic enzyme, sorry. And vice versa, those population with a low frequency in their target site mutation had higher enzyme activity. So, and that's to show that these mosquitoes were all taken from St. Andrew, Jamaica. So it shows that we cannot just design an insecticide for a particular target site. We also have to look at other factors in how the mosquitoes adapt to become resistant to the insecticide. So we have to look at, is the insecticide penetrating the cuticle? Are they affecting the metabolic enzyme? Are they suppressing enzyme activity? And can they bind to the target site? So we have to design an insecticide that has multiple modes of killing the mosquitoes for it to be effective. The other study that I conducted between the period 2018 to 2019 was I looked at a larger area of Jamaica. So I screened mosquitoes from the eastern parishes of Jamaica, particularly Aedes aegypti. And I'll show you the trends in that. So I'm only presenting, in this case, I'm presenting on St. Catherine, Jamaica, but all the trends were similar throughout. So I've just taken one parish and just presented the data here. So I'm looking at the family of Pyrethroid insecticide. And there are several insecticides family. There are pyrethroids, there's the organophosphate, there's the organochloride, and there are bendiacarb. So 4 known, and they're still developing more. So from the family of pyrethroid, I tested 4 insecticides in that family. I tested the concentration at the diagnostic dose for resistance at the ×1 in blue, ×5 in red, and the ×10 in gray. Only 1 insecticide was able to kill 100% of the population at its highest concentration, that's ×10. So, and the others had little to no effects. Major activity was below 80% of the population died. So that shows that only deltamethrin was effective at killing the Jamaican population at its highest concentration. And we have to be careful of how high we go in the concentration, because us humans will be starting to be affected by the application of these insecticides as well. Now, to reiterate the point that we need to do more in finding out about our first line of defense and whether they work or not. From my report in 2021, I looked at all the countries that were reporting evidence of insecticide resistance, and consistently it appears that in the recent time between 2000 and 2020, only Guyana, Guadeloupe, the Grand Cayman, Jamaica, and Martinique were the countries that have recent evidence of whether their insecticides are active or not. Okay. So other trends. So I've shown you that insecticide resistance is a problem. I've shown you that there are problems with the containers. Because of increasing container use, the mosquitoes have become drought resistant. So I'm now looking at our habits and what's affecting them. So forest encroachment is another habit that we need to be concerned about. Some people go into the forest for hunting, for farming, and also for housing development. That causes a problem of transporting a sylvatic or forest virus into an urban cycle. So the Mayara virus, I'll use that as an example. The Mayara virus was first observed in the 1950s in Trinidad in its farm workers. Farm workers coming out of the forest were observed to have this new infection. And with this, they detected that this new infection, of course, is the Mayara virus, and they named it. Since then, they've noticed that the Mayara virus is localized to South America and Caribbean countries in close proximity to the South American border. So below Trinidad, Guyana, Suriname, French Guyana. Now note that Mayara virus within the forest is transmitted by the Haemogogus mosquito as well as the Mansonia. However, it has the capability of being transmitted by the urban Aedes aegypti. So with increased forest activities, all it takes is one viremic individual to be bitten by a local Aedes aegypti for that pathogen to be transmitted in the urban cycle. Also to note is that in 2016, this virus was observed in Haiti. Now, Haiti is in the Northern Caribbean country, it's a Northern Caribbean country. So the virus has increased its geographic range from South America or Southern Caribbean countries to North Caribbean countries. And most importantly is that the virus has mutated. So there's now a Southern Caribbean strain, and there's also the Haitian strain. So we have to be careful of how we further conduct activities for housing development and farming, because we stand the possibility of bringing pathogens that normally circulate or cycle in the forest into our urban setting. So I also said that with this, I'll be talking mainly about mosquitoes, but I'll be including other insects that are showing similar trends and patterns to that of the mosquito. So these are triatamine bugs that are normally found in the Caribbean and South America. Triatamine bugs transmit Trypanosoma cruzi, a pathogen that causes Chagas disease. And Chagas disease causes enlarged hearts. So in about the 1960s, studies from individuals in Trinidad that presented with enlarged hearts were tested for the antibody Trypanosoma cruzi. And what they found was that these individuals were indeed positive for Trypanosoma cruzi. Around that period, other studies were conducted in Aruba and Curacao looking at farming homes, and they collected the triatamine bugs in and around farm homes that were close to the forest, and they found that the triatamine bugs were indeed positive for the antibody Trypanosoma cruzi. However, the individuals living in the farm homes were not infected by the pathogen. Later, from the 1960s, 1999 thereabout, Belize conducted a study looking at communities in close proximity to the South American border. And what they found was that the triatamine bugs collected from those areas were infected with Trypanosoma cruzi, and about 6% of the population in that area were infected by the pathogen as well. Belize went a little bit further to look at the urban areas and collected bugs, triatamine bugs in the urban area, and they were also infected with Trypanosoma cruzi. So as I said, the triatamine bugs are normally found in forested area. And they're important to South America and Caribbean countries close to the South American border. So in 2014, Grenada decides to conduct a study on triatamine bugs found there. And they conducted a study looking at 399 dogs. And they found that of the 399 dogs that they tested, 19% of those dogs were infected with Trypanosoma cruzi. And of the 19%, 6% of the dogs were domestic or pet dogs. So the only way that pet dogs can be infected by a pathogen that's normally circulated in insects in a forested area is one, if the insect is coming close to urban area as they found in Belize, or the owner is taking them into a forested area. One out of the two, whichever the case is, we need to look at how we conduct activities in and around forested areas. Another pathogen to note is the one that transmits Mansonelis by the Culicoides species. And Mansonelis is mainly important in South America. It's actually transmitted by the black bugs or Simulides. But in Haiti, it's transmitted by Culicoides. So this is a pathogen that can be transmitted by two different genus or two different types of bugs. And in Haiti, because the pathogen presents with mild to no symptom at all in the population, even although it's prolific, it goes unreported and untracked by the local health authority. A pathogen to be concerned about that has been adapted to temperature is leishmaniasis. Now, leishmaniasis is transmitted by the phlebotomine sandfly, a sandfly that feeds on blood. And we have those vectors throughout the Caribbean. Now, in 2014, an interesting report was made. So prior to this study, leishmaniasis in the Caribbean was observed to cause cutaneous infection. So it causes lesion on the cutaneous membrane, so on the skin only. Leishmaniasis minder is able to present with three different symptoms. You can either have the skin infection, the subdermal infection, or organ swelling, which is visceral infection. So in 2014, a common pathogen that normally is observed in immunocompromised individuals, those with HIV/AIDS, were shown to have symptoms of visceral leishmaniasis. So here is this pathogen, Leishmania martiniquez, that normally causes skin infection, causing skin lesion, now infecting the organs of the body. And in Martinique, they speculated that the reason the pathogen was travel from the cutaneous towards the viscera is because of changes in ambient temperature. Now, again, I'm asking you this question. Which part of the body do you think would be cooler? The skin or the internal organs that are surrounded by fluid? My guess would be the internal organs that's surrounded by fluid. So here's this pathogen that's modifying its behavior from being predominantly skin infection to now causing organ swelling and targeting the viscera. So the pathogen has become temperature sensitive. So with all these pathogens and organisms that are presented and their trends, I think that to move forward, we need to learn from the Zika virus. We need to understand that for us in the Caribbean, because we're so heavily dependent on trade and travel and tourism, that all it takes is for one viremic individual to enter into our country. And we have loads of insects. We have an abundance of insects. So all it takes is one competent vector, insect vector, that's able to bite this person and transmit the infection locally. And then because of our trade throughout the region and travel, for the pathogen to travel or spread throughout the entire region. The other thing that we have to notice with Zika, when Zika was first observed in about the 1940s in Africa, there's a possibility that no pregnant woman was involved. Studies have found that the pathogen, the virus, mutated from infections in Africa to infections in Asia, and the strain that we have that's transmitting in the Caribbean and South America is very much different than the strain that was originally discovered. And maybe with that mutation, we're seeing all these new symptoms coming about. So it's absolutely important for us to continue with aggressive vector control methods. Yes, it's expensive, but we have to do continuous vector control and epidemiological surveillance. Epidemiological surveillance is also important as vector surveillance in order to have an idea of what is circulating in the population. And we're not doing much of that. We cannot mitigate against the unknown. We have to know what's out there, and in having a grasp and understanding of what is out there, then we may be able to better equip ourselves with something else that's coming in. We know that we have competent insect vectors throughout the region. And because of our activities in travel, we're getting more pathogens coming in. Zika and chickpea is one. Also, I mentioned to you before that the mosquitoes, looking at the mosquitoes in particular, the mosquitoes, they go through their phase where they don't need water. The eggs don't need water, so you can transport one organism from one country to the next. And we're seeing the introduction of species like the Aedes albopictus, which is another competent vector at spreading dengue, chikungunya, and Zika as well. Water restrictions do not affect the insect. Our container activities are ensuring that insects, these mosquitoes, are drought resistant. Deforestation is a problem because we stand the possibility of introducing a forest pathogen and or insect into an urban setting. The insecticides have minimal effect, so we need to find out what works for us, and that consists of consistent insecticide resistance testing. And temperature is a problem. Our increasing temperature that we're observing, the mosquitoes have adapted to it in that they're decreasing their time to adulthood. And we see now that pathogens with Leishmania have adapted, wherein they're no longer infecting the skin, they're going towards our organs. And it's also very important, because of all these changes that we're seeing, to equip ourselves with novel tools to manage our vectors. So while we're sitting here trying to find out what is it we're going to do in our ever-changing environment, we must bear in mind that the insects have adapted, the pathogens have adapted. So it's us now to catch up. Thank you very much for listening. I will take any questions at this time.
Thank you, Sheena. Any questions either in the chat or live? Nothing from the group so far. I have a quick question for you, Sheena. With a lot of the ridge to reef concepts coming to the to various SIDS, the importance for water resources as well. Some of these ideas are that for farmers, or so say farmers on the ridge, to actually try and conserve water, putting check dams in, or water retention ponds for irrigation. How do you educate farmers on that? It's kind of a two-edged sword, where we need water to irrigate, but of course we have all these different insects that like water to
populate.
Grow in.
Yes. Well, it's It's management. At the end of the day, it's all about management, and it's difficult, but vector management has to take a community-wide approach. We can't just depend on the health officials to conduct vector surveillance or epidemiological surveillance. We have to take it to the community level as well. So in harvesting water, we have to be very careful of well aware if we're seeing any larvae in the water, because there are some population of insects that can breed in salty and brackish water. And we need, if and when we see that, then we need to involve and include the health officials and ask them what can be done. So we can't work in silos anymore. We have to be inclusive.
You bring a good point on water harvesting. Everything I've seen in the islands, most people that do collect water, they are covered. Is that correct? Or is that something that's being taught throughout the region?
It is being taught throughout the region. It's being taught to cover the water. That's a recent activity because of the increase in vector-borne diseases that we're seeing. So individuals are being educated by their health officials to cover harvested water. But besides that, there are still many containers that are uncovered, or the covering is compromised. So they might use a net, and the net has large pore holes to allow the mosquitoes to enter nonetheless. Or the net might be touching the water, so the mosquitoes can actually lay on the net, and because it's touching the water, the eggs get in, and all they need is a hole to escape, or when the person is lifting the cover for the mosquitoes to escape.
Thank you. Any questions from the group? It's like we have one question here from Doug Evans. As insecticide resistance increases and newer products are developed, are pesticide toxicity risks to humans are there any pesticide toxicity risks to humans or other mammals? Are they increasing?
So all organisms have enzymes and specific target sites that exist for a chemical or a compound, and majority of these insecticides that are being designed now, they do target either an enzyme or a chemical target site. Similar, for example, I look at malathion. Malathion targets the acetylcholine esterase enzyme, and these enzymes can be found in the respiratory tract of humans. So some people, whenever insecticides are being sprayed, they find that they cough. So yes, the answer is yes, the insecticides can affect human, and that's why it is concerning. Resistance is concerning, because if we, instead of designing new insecticides, if we just continue to increase the amount of insecticides that's being applied, then we stand the possibility and chance of affecting other organisms, including humans that are around.
That's interesting. Just even with development of new, so that's an OP organophosphate pesticide in the pyrethroids. But if you look at South Africa as a case, back in '99, they dropped using permethrin and moved back to indoor spraying of DDT. Oh, part of the reason was through resistance, and the other was it was expensive, and DDT seems to still work. Is there any regions within the Caribbean that are bringing back DDT as a solution, or has this totally been banned, say through CARICOM countries?
Not to my knowledge. I don't know of any country that is bringing back the use of DDT. I think based on the WHO policies that I've read, that DDT use is restricted to African countries because of malaria outbreak.
Right, correct, yes. So another question, just maybe a couple more from Adrian Cashman. Have you focused on the adverse effects of mosquitoes So I wonder if we are able to eliminate them using some of the interventions in the reproduction cycle. Would that be a good thing?
So thanks very much for that question. So there are insecticides that are now being produced that targets the ability of the female to lay eggs and reproduce. So these new insecticides are in development at this time. So there are insecticides that prevent females from actually taking a blood meal. And there are insecticides that prevent the females from actually laying an egg. And there are also growth inhibitors. Those have been around for a while though. Growth inhibitors prevent the mosquitoes from progressing on to their adult stage. So they'll retain or stun development to the larval stage, and the mosquito will stay in a larval state until they eventually die. So there are novel insecticides that are being produced at the moment.
Thank you. And to follow up, what good do mosquitoes do in the environment?
Mosquitoes
That's a common question.
Mosquitoes are pollinators. They pollinate our plants.
Yeah.
So
yeah.
I got the question before from some of my students as well. So why not? Why do we need them if we have bees? And if you're going to ask that, then the answer to that is that sometimes we have very, very small plants that the bees are too large for. So we need the smaller insects like the mosquitoes to pollinate them.
Yes. Well, great questions. I think that, are there any more questions from participants still online? If not, Sheena, I would like to thank you for an excellent talk. I learned a lot at this one. So thank you so much.
Thank you too. And it was nice for you to have me.
Thank you. Before we close, everyone that's still on, next week on November 9th, we have the 8th talk in our series, wastewater. Rest of your week. Thank you, Sheena.
Okay, thank you again.
Cheers.